And I'm start sharing my screen. Okay. Pretty good. So we covered more or less everything about DNA and RNA. There is just a few slides that I will go back and describe RNA-seq for you guys. If you have went through the bulk RNA sequencing, it would be easy now to understand how we can process RNA sequencing, and then we will move to the proteins later on. So the main body of this lecture is going to be discussing the proteins. And there was just only the single cell RNA sequencing, which we didn't cover in the previous lecture. Now, I understand that sometimes when I'm describing for you DNA sequencing, as I mentioned, it's something that is really expensive. It needs a lot of expertise, and it's not possible that you go yourself to perform RNA sequencing for a whole genome. But it's important to just get exposed to how these things are done. The same thing as I mentioned in terms of the RNA analysis. So if you get to any lab and you are gonna be exposed to some experiments or perform an experiment, then you will be able to isolate the RNA, probably perform some qPCRs, maybe microarrays at maximum, and then RNA sequencing and single cell analysis, you will need to do that during your PhD. Like you have to get already to that level to be able to perform those. This is because these are very expensive. So your supervisor is not gonna let you perform a test experiment on RNA sequencing, which costs around $10,000 per experiment, or single cell analysis, which costs around $30,000 per experiment. So really expensive. So from this lecture on, we will move to the protein analysis. And just to remind you, we discussed how we generate the total amount of RNA. We isolate this from cells, and then we generate a library. And this library can be either fragmented at the level of RNA or cDNA. And then we have short repeats of the RNA that we can sequence. And then I've shown you that there are multiple platforms where the RNA sequencing take place. There are different ways to prepare the samples for those. We are not going to these details. But the concept of the RNA sequencing is more or less the same. So the flow cells that you use for a bulk RNA-seq or an RNA-seq from the whole tissue that we described, we've seen the videos. We really stressed on using Illumina because it's one of the most popular. We have here platform in the OHRI General Hospital where they have multiple Illumina sequencing machines, and it's fast. And we've seen how it works, and we've seen the comparison between RNA sequencing and microarrays and the caveats of which one to use. Now, in the preparation of the RNA sequencing, there are a few steps that you really need to know because these are very basic. We are not really diving very deep, particularly when we generate these 300 base pairs, adding the adapters, then adding the primers. And the end product, it would look like a small piece of RNA, a cDNA that has sites that allow binding to the flow cell. And that's how we get our RNA, or in other words, the cDNA attached to that cell. And we can, in principle, sequence it and get the information out of it. So the endpoint was, if I'm comparing a whole tissue that you got from the pancreas of a healthy subject and a pancreas of somebody who had a tumor, then you are taking a group of cells. And we mentioned that when you take the group of cells, then you are getting all the transcripts from all the cells. And therefore, for this red transcript, you will get the average from all the cells. So maybe there's 5 red transcripts from this cell. There is 2 red transcripts from that cell. There's 15 from that cell. So we get an average from the whole cells together from one tissue compared to the other tissue, which is not presented here, like if it's a healthy or tumor. So this is already good because it gives us a good overview of the difference between a healthy subject versus a diseased person. Now, or a diseased tissue, sorry. Then we can get the averages. So we can say that there are 5 samples here. We could already see that the first 3, they have high cell cycle expression, and the last 2, they have low cell cycle expression. You can get all these correlations, and you can get the single gene if it's up or down regulated. You can clearly get those numbers. But now technology is advancing, and we are at the level where we can do RNA sequencing on single cells. So instead of taking the tissue altogether and comparing it to another tissue, which is not presented in this image, like again, healthy versus diseased, then within this particular tissue, you're gonna dissociate it first into single cell suspensions. So you will take the violet versus pink versus green cells. And for the violet cells, you're gonna extract the RNA, cDNA, messenger RNA, cDNA, and then for each one of those, and you will compare the single cells on this tissue to the single cell components on the other comparative tissue. And the end result would show you a map like this. There are multiple formats of these maps. You might see here TSNE, or you can see UMAP. It depends on the software you're using. But then you get clusters of those cells. In other words, these cells, they are transcriptionally similar, and those cells are transcriptionally similar, and those are transcriptionally similar. But obviously, the yellow cells are transcriptionally different from the green cells. And of course, if you get more detailed in the analysis, then you can go and say, what happens to those cells? Do these cells become those? You can ask questions like that, which you cannot do with the bulk RNA-seq. So the principle is the same, but the sample preparation and the procedure is different. So in terms of the RNA sequencing part, it's almost the same, but how to get there, it's different. So in the single cell analysis, it provides you unique transcriptome analysis or landscape from individual cells. So you have to take those cells and sort them out. If you are, for example, I'm using the heart. I'm going to show you just an example in the next couple of slides. So there are 4 major components in terms of cell types. There's like fibroblasts, endothelial cells, leukocytes, and cardiomyocytes. So then you can sort these cells based on their surface markers. And then you can get each of those cells barcoded. And the barcode is, again, it's a DNA sequence. And you make libraries. And after that, you can get to these results. Obviously, there's a high-end level of bioinformatics that you need to know. Like our programming is very well needed, if I want to say. If anyone needs to do single cell analysis or go bioinformatics, I really advise you to learn our language. While on the bulk RNA-seq, yes, you get that average, all the cells are mixed together, you get all the RNA, and then again, we are doing the barcoding, but the barcoding now is for the whole tissue together. Yes, you will get a differential gene expression, but it's an average of this tissue compared to another tissue. And that's why RNA-seq is much more, single-cell RNA-seq is much more powerful, and it's more and more used these days in molecular biology. Like if you look any publications, you will see like single cell analysis there. So how do we prepare the sample for the single cell analysis? First, we have to get the cells dissociated into single cells, and we have to maintain them alive. So this is really very difficult because many times when we manipulate tissues, these like imagine you have the heart and you are dissociating it into different tissues. So you have to do digestion steps. Many cells die. And there's also flow cytometry where we have to separate the samples as I showed you here. That is really a very harsh technique on cells too. But then imagine you have succeeded in this and you got the cells in suspension. Then we perform the platform I'm showing you right now is the 10x Genomics. It's called 10x barcode here. The company is called 10x Genomics. There are others, of course. The way it works is each cell, so here there are representation of 3 different cells. So each cells is encapsulated with a barcoded bead. So these beads, they have a barcode on their surface. So the barcode of this green bead is different from the barcode from this violet one and different from that orange one. Now, when this cell and this bead are together, they can give me a whole separate sample because whatever RNA is going to be expressed on that cell, then it will have a different transcriptome. And I would know the transcriptome based of that cell being with that bead. So there is a way to retrieve the information and it's represented here for you. So if you look at the surface of the bead, there are oligo-DT. So remember the oligo-DT primer for the reverse transcription. And there is this sequence here, which is called the 10x barcode. And the 10x barcode is simply a unique DNA sequence that would identify this bead from any other bead. And therefore, if you get this cell, you get the transcriptome from that cell, you would know that the transcriptome comes from this cell because of that bead being together. After that, we will generate a library, and I will show you in the next slide how the library is generated. And then we go to the steps that we described before with the flow cell of the Illumina analysis of sequencing. And after that, there is the bioinformatic analysis where we can dissociate cells from each other based on their transcriptional profile. So the process of how I can make the beads and the cells together is very, very tedious and very, very precise. And this is the most important step in the experiment. So first, we took the samples and we dissociate them into single cells. And after that, I took those and I inserted them into the 10x machine. The 10x barcoding machine, it works like you have one sort where you get the beads, and from the other side, you are getting the cells. So when you insert the cells. So they are running in such a simultaneous speed so that one cell can meet at this junction with the bead. And then as they are flowing through, there is oil that is being added here, and the oil will make it droplets. And the outcome is probably sometimes you will have only a cell in that droplet of the oil. Sometimes you will have only the bead. So they are showing you one example here where there's only bead, no cells. It could be that you get a cell with no bead, and there's a chance where you get the cell and the beads together. So the efficiency is really high. There is 20% loss of the sample. So 80% of the sample will probably have cells and beads together. So now you will have within this oil droplet only one bead and one cell. Within the oil droplet, you will have also reagents. Those reagents, you will have the reverse transcriptase, which will allow the reverse transcription and the generation of the cDNA library. So they are showing you here the outcome. Some oil droplets, it has only the reagents, no cells. Some of them, they have beads only. Some of them have beads and cells. Now, the successful ones here, is beads and cells, where it goes to a next step where the lysis take place, and then the hybridization of the RNA to the oligo-DT, which is shown here. It happens on those beads. And after that, you go to the process of the generation of the cDNA and the sequencing process. So there's a lot of complicated steps here. I don't want you to know. If you are interested to read more about how these things are done, there is very good references for you here, but it's not really required from you. Just for you, if I didn't mention before, the UMI, what it is, it's a random sequence that is added on top of each of those beads, and it's unique for those beads. Now, say we have done all that, then we are able to retrieve all the sequences, and then we perform the analysis and do a differential expression between two different populations. So this is one of the experiments I am doing in the lab. I actually have done in the lab in the last year. And I'm trying to study the transcriptional profiling in the heart during healthy conditions and in diseased conditions in mouse hearts. So I did enzymatic digestion where I dissociated the cells. I perform antibody analysis to staining so that I can differentiate the 4 different cell types in the heart. There are 3 represented for you here, and if you will get a little bit confused on this slide because of antibody staining and the flow cytometry, these 2 techniques we're gonna describe in the next couple of lectures or so. So it's okay if that doesn't look very clear for you if you have never heard about antibody staining. The bottom line is that you use a specific antibody that detects a specific cell type. We know that CD31 proteins are only on the surface of endothelial cells. So if we have an antibody that is detecting the CD31 positive cells, then these are endothelials. We know that CD45 is a surface marker of leukocytes, which are the immune cells. And therefore, if you have an antibody that detects the CD45, then you can capture all the leukocytes. And the fibroblasts are negative for both. So this is the component where you see here. So I did experiments where I isolated cells from healthy hearts and diseased hearts, digest them side by side, stain them side by side, and then I perform flow cytometry to get these cells separated. And then the same as I explained to you in 10x genomics, we ran the barcoded beads with the cells, and then we get the oil droplets, and then we can collect the cells and perform the RNA-seq on each particular cell. Now, you can imagine that this is very sensitive because the amount of RNA that you get from a cell is very little. So it's super sensitive and super accurate. Now, clearly you can tell in this experiment that the transcriptional profiling at the level of cells, so there are multiple populations in the heart here. You can see the one, I just pointed 3 out of many. So you can clearly see that this pink population here in healthy heart kind of disappear in diseased heart. So this is an indication that those cells play a role during the cardiac disease. You can see that those cells totally disappear at all. So they are wiped out from the heart. So probably the function of those cells is important to adaptation of the heart function. And what I found was these pink cells, in fact, a transition to become those cells in a diseased heart. So when I followed the trajectory of what those cells become in diseased heart, I found out that in diseased hearts, the pink cells become those orange cells. Then you can ask particular questions that are very specific. Imagine you want to study a gene that you already know that it probably play a role, and you want to say, oh, where does this gene is expressed in the cell population. So here's an example showing you that I clearly could detect that this cluster here in green is very abundant for the expression of that gene, and most of the other population don't. So this is the type of data quality that you don't get with RNA-seq, because probably there you will see the expression of the gene X, but you wouldn't know which population of the cells. And now, of course, we have for each of those clusters, there's a lot of gene markers that I can check. So I would know what exactly this cell type is and what exactly this cell type is. So this is really a very, very high-end type of analysis. Now, obviously, single cell analysis can give you so many information. And the information you can get is, as I mentioned, you can ask questions, what does this cell become? And I gave you an example here that I know based on the transcription profile of those cells. With stress, they still keep some of their gene expression profile, but they really become that later on. So these questions are very normal to be asked in such analysis. And obviously, you can take healthy subjects or pathological subjects now, and then you do the RNA-seq on single cells. And if there is a population of cells that is very, really, really rare, that most of the time is masked by the populations of other cells, you can clearly identify that. You can probably say, oh, in healthy subject, there is like these three cell types, which I can see they are also in the pathological condition. But here is a cell that shows a totally different transcriptional profile that is only present in the pathological condition. So therefore, these cells could be either adaptive for the disease or they are the cause of the disease. And then you can start to ask all these questions that are raised down here in the slide. So the bottom line, single cell analysis is more specific, more powerful, and the amount of data you get is amazing, huge. So in the example I showed you here, each single dot is a cell. So I analyzed around 80,000 cells. So in your Excel sheet, you will have 80,000 cells with the 20,000 genes or so. So the amount of data you get is amazing. Okay. Now, you can perform also single cell on tissues as well. So if you have, for example, here they are giving you an example of the brain. Forget about everything in this slide here. I just didn't want to modify it, and I wanted to keep only that part to tell you, you can take a small part of a tissue and you perform single cell within the tissue. So you can also get the transcriptional profile of each cell, but also its location in the tissue, where it's like located. So merging these two together would tell you, for example, if those cells who cause the disease are migrating from one side to another, or they are located on that side or another in the tissue. So that's special studies that are merged between single cell and RNA sequencing. Okay. So I hope this gives you an overview of the single cell analysis. And with this, we are closing the chapter of the RNA-seq, and we will move to studying proteins. Now, to prepare you for studying the proteins, there will be 3 or 4 lectures about how we isolate the protein, how we analyze the protein, and what are the major techniques that we use. And then we will move to describe 4 or 5 applications that we use, like when you go to do blood test, when you go to your urine test, and all these things are in principle detecting certain proteins. So first, we are already familiar with isolation of proteins. We discussed how we homogenize a tissue or cells, and then how we isolate the proteins. Depending on the protein types, we will be choosing whatever buffer is needed. Then the second step would be to understand the basics of the protein structure and particularly the antibodies. So antibodies, exactly like sequencing has shifted the molecular biology in terms of DNA and RNA and our knowledge. Without antibodies, probably the molecular biology techniques would have been really, really difficult to understand protein function. So most of the techniques that we use in the labs are based on using antibodies. And obviously, when we say most of the techniques we are using need antibodies, this means we are generating antibodies that are specific to certain proteins. So in other words, today I discovered a new gene. I did RNA sequencing, cDNA or RNA sequencing or genomic sequencing, and then we discovered there was a gene that we have never studied before. We know that this gene has a promoter and it's producing an RNA and it's translated to a protein. So if I want to study that protein, then I would need to generate an antibody against that protein in order to be able to detect it. So we will describe in the next lecture how we generate antibodies and how do we use them in research and in clinics. Antibodies, not only used in research, we could use antibodies to sequester certain diseases. And I will give you some examples as we go in the next lectures. So after that, we will dive into the techniques that we do in the lab, like immunoassays, like Western blotting, immuno-microscopy, and immunoblottings, and the fluorescence-activated cell sorting. This is the one I just showed you in the previous slides here, where we isolated the cells. So we would need antibodies against specific surface markers to identify cells. So I will describe that as well. And immunoprecipitation. So for example, if you have one protein that you would like to see with which other protein it binds. So you have to also use everything we're going to use here. It will depend on the generation of antibodies. So I will go quickly on most of these slides because we have probably visited those slides, but it's for you as a revision and to set the ground that probably you are already forgot what we have done before the midterm. So I mentioned many times that proteins are everything in terms of function of your body. If you look, there are a short list, I want to say, of the total functions that proteins perform in the body. Like enzymes are proteins, hormones are proteins, signaling between cells and within a cell are proteins. Membranes, they are mixed between proteins and lipids. Antibodies are proteins, which are the most important in the defense against pathogens. The organization of different cellular processes, including transport, like insulin transport, glucose storage, opening and closing of channels, regulation of acid and base, and the amount of water, the fluid balances, all that is done by proteins. Of course, these proteins might be associated with other molecules, but without proteins, all these functions are not going to be there. So that's why the end product of any study that we perform in labs and that we would like to take into clinic should have for sure to look at the changes that happen at the protein level. So when we speak about COVID-19 generated antibodies, this means we are speaking about what are the antibodies that are able to sequester the COVID-19 when it's coming to our body or when we get infected. if you like, I'll give you a lot of examples as we go through, but this is just a light introduction now. Okay. Now we described how we generate a protein, and we kind of get into the details where, of course, DNA sequencing helps us to know the sequence of the gene, and RNA sequencing can help us to know the messenger RNA that is going to produce the protein. And in other words, the sequence of the protein is kind of pre-known because we know the RNA and the gene that produced that protein. So the sequencing of the RNA, I'm not gonna of the proteins, I'm not gonna really focus so much because nowadays we would really perform the recombinant DNA technologies to generate any protein that we want. All we need is just the cDNA. Only in conditions where we are describing a new protein that you have never studied before, then you would do protein sequencing, okay? So we discussed that a gene is transcribed into RNA, and the RNA is translated into a protein, and we discussed this in module 11. If you didn't go through that or you forgot it, please go back to that, these two slides. And the end, we will have the generation of these polypeptides, and the polypeptides will generate the primary structure of the protein. Now, the protein, as I said, as a sequence is known, but also it has a directionality, and the directionality comes from the fact that it has an N-terminus and it has a C-terminus, and the amino acids are changed from one another based on the radical group that you have here. Whenever you add an amino acid to the peptide, it would be that added to the carboxyl group here. So that's what the peptide bond that you will see generated here. Okay. And therefore, so we know the sequence of the amino acids based on the RNA and the DNA, and we know that there is directionality of that protein. Now, the primary structure is just the sequence of the peptide, the amino acids. The secondary and tertiary structure is the folding of those amino acids. Obviously, the R, which is the chain that is added on different amino acids, it could be positively charged, it could be negatively charged, and that would create different stoichiometry of repelling those molecules from each other or attracting them to each other, and therefore you start generating those secondary and tertiary structures. Most of the time, the tertiary structure of a protein, if it's combined with other peptides or polypeptides, you would generate a functional protein. Like here, it's hemoglobin, where you have 4 different components of polypeptides. And in many times, proteins don't work only on their own, so they form protein complexes. So there is, in this condition, there is only hemoglobin here. But if you would look to any signaling molecule, this protein probably will bind to a receptor, and the receptor is also a protein. And then the receptor will have some changes, and there is a lot of downstream changes that happen in the cell. So after the protein is generated, there are some post-translational modifications that take place. And if you study the post-translational modifications, this would add one more level of complexity to the genome itself, because recall that we have 20,000 genes or a little bit more. And then the first level of complexity is that we are generating multiple RNA transcripts from the same gene. So you remember IGF-1 that you did in assignment 2, it has probably 7 transcripts. 2 of them or 3 of them generated proteins. So from the same DNA sequence, you are generating 3 different RNA transcripts. And then if you generate these 3 different RNA transcripts and you translate them, they will be 3 different proteins. And if you add those post-translational modifications to those proteins, you will add one more level of complexity where you can activate a protein or deactivate a protein. The most known and the most studied, at least at your level when you get to your master degree, are the phosphorylation of a protein, acetylation of a protein, making a disulfide bridge between the proteins, or hydroxylation of a protein. So I focused on, I wanted you at least to remember that there are multiple post-transcriptional modifications. Acetylation, phosphorylation are mostly studied, and I'm giving you an example here of two different enzymes, which happen to be also proteins. So the kinase is an enzyme and it's a protein, but the function of that protein is to phosphorylate other proteins. And the phosphatase is also a protein, which is an enzyme as well, that dephosphorylates other proteins. So it has a function to add a phosphate as a kinase or remove a phosphate as a phosphatase. And the outcome here, you can see that phosphorylation itself, when you add a phosphate group to a protein, it can either generate that, it causes that protein to become more active or inactive. So in the orange, it makes the protein active. So normally this protein is inactive. It cannot perform a job. The kinase came and gave a phosphate group, then this protein becomes active. So this is how you can control the function of proteins only by post-transcriptional modification. Or you can do the other way around. Probably this protein is already active in green. Now you add the phosphate group and then you can block its function. When the phosphate is bound to that protein, it's inactive. So there are a lot of these examples. And these processes are elegant and so quick. Phosphorylation events, it take less than minutes. So if you treat cells with, let's say, TNF-alpha, which is a cytokine, within 5 minutes, you have most of the targets of that protein phosphorylated. So this is really, really quick. And that's why the cell decides to have different levels of controls. And this control is not only elegant, it's also very quick. Now, we discussed before the cell disruption and fractionation in module 3. And we discussed that depending on what is the outcome of the experiment you are doing or the question you are asking, you have to make sure what type of buffer you're going to use. And you have to also make sure that you are doing this in cold condition, particularly because most of the enzymes are not active when they are at 4 degrees, like proteases. These are enzymes that break down proteins, and RNases, these are enzymes that break RNA. So these, if you put them at cold at 4 degrees, they will not be very active, and therefore you can maintain the protein pool you are extracting from the cells or the RNA pool you are extracting from those cells. Then we also discussed that depending on where you are extracting proteins, you will decide on the type of the detergents you're going to use. So in this condition, if you want to use denaturing reagent, the most used is the sodium dodecyl sulfate, which is mostly known as SDS. And if you want to use non-denaturing reagent, this would be non-ionic or reagent like we discussed about Triton X or MP40 or Tween. So this will be little bit less harsh. So if you are extracting, for example, a protein just to do Western blotting and check how much of that protein is, you don't care about the function of that protein. You just care to see if it's there or not. So you would go here. But if you are extracting an enzyme and you want to see the activity of the enzyme, so you don't want to kill the enzyme, so probably you want to go this route. So depending on the experiment, you would choose what to do. And obviously this comes from experience. Most of the time you would look what other people published and what techniques are used, or you can consult companies that sell those products to give you a better outcome of what to do. For the Western blotting, most of the time, if you want to check the existence and the quantity of a particular protein in a cell, most of the time we use the SDS because we look at the protein at its denatured state. Now, the most widely used buffer is RIPA buffer, RIPA, and the components are there. We discussed that. You have to be careful that the RIPA buffer needs supplementary components like proteases and phosphatase inhibitors. So you want to kill those enzymes. Otherwise, imagine you are looking to check how much of this protein here, active protein. This active protein is phosphorylation, is already bound by a phosphate, right? So when you extract the protein, you really want to maintain that this protein bound by phosphate. Otherwise, you will be taking the inactive form of that protein. So if you are extracting a protein where you want to check the phosphorylation, you want to make sure that you don't have phosphatase. You want to kill the phosphatase enzyme. Otherwise, the phosphatase will come and remove the phosphate from your protein. So these things you have to take into consideration when you choose your primers, your buffers. And obviously, if you don't put protease inhibitor, you will degrade your proteins. It will be destroyed into small peptides that you cannot utilize. So be careful what you use and how to use it. And these slides already discussed that for you. And the example I mentioned before, how to use the cells and how you can collect your cells in RIPA buffer. This note here to remind you the choice of this lysase buffer and protein extraction method, it depends on which experiment you're doing and what's the aim of the study you are doing. Okay, so what's next? If we are discussing about the total amount of proteins, and we know that proteins, they form different structures based on their primary sequence of amino acids. So the primary structure is this sequence of amino acids that form. Here's an example of alpha helix or beta sheets. I'm not really going into the details of the structural biology here. And then this folds more, the longer this peptide will be to generate the tertiary structure, and probably make subunits to generate the quaternary structure of a protein. Now, back in the time when people were starting to find ways to sequence the biomolecules like the DNA or the RNA, they also thought about sequencing the proteins. And I'm not going to the details of this because it's very rarely used unless you are really a specialized lab of biochemistry that performs sequencing for proteins. The idea is very simple. You would label the first N-terminal amino acid, and then you cleave it, and then you can detect it by chromatography. And you keep doing this more and more till the end of the protein. This is a very laborious process. You need a lot of proteins for that. Now, there are other aspects that we could study which could give us an information about the secondary and the tertiary structure. Now, the first data set you can get of a protein is probably based on the RNA sequence. But if you don't know where this protein comes from, then you don't have a choice except by sequencing. So that will give you the primary structure of the protein, or in other words, the sequence of the amino acids from the N-terminus all the way to the C-terminus. You could also do that by bulk analysis, where similar to how we performed the bulk RNA-seq and or bulk sequencing of the DNA, you would isolate the proteins from tissues or cells or tissues from different conditions like diseased or not. Then you would run those on a gel, and the running on a gel, we will have an introduction next lecture, how we generate these gels and how we run the proteins in a Western blotting setup. And after that, you can digest those. Again, if you every single time we do bulk analysis in DNA, we had to fragment the genome into smaller pieces because the DNA genes are really big or long. We used restriction enzymes. With the RNA, you remember we also fragmented the RNAs into 50 to 300 base pairs. And again, here you are fragmenting the proteins into peptide. And for that, we use the enzyme trypsin because it's known where it cut and it generates peptides that are relatively short that we can detect. And then we could use the mass spectrometry, which mostly used is called MALDI, which based on matrix assisted laser desorption ionization. Obviously, I have never done this in my life with all the experiments I did. Yes, I used the core that performed that, but I never did in my life an experiment where I operated this machine, and none of you is going to do unless you have many, many years of experience. Or by electrospray ionization. So this is a different way to ionize those peptides. In other words, giving them a positive or a negative charge, and then separate them based on either their molecular weight, so how many amino acids there are, and what is the charge, so the mass and the charge. So you get different peptides, and you would know what is the size. To know the peptide mass by fingerprinting, you should already have an indication about that protein or peptide. So in other words, you have in the database the protein sequence, and you are just matching how much you get out of it, like how much of that peptide you get in the analysis. Like now, for example, we have 600 different kinases. If you are looking for one of the kinases, we know the sequence, we know the amino acid sequence. So if a peptide like this one or that one or that one, based on the how many amino acids and what is the charge that is showing up, we could easily, based on the sequence, say, oh, the amino acid, let's say here, phenylalanine, leucine, serine, and cysteine, it comes from this protein. So I can locate it back to which protein it comes from. Now, if I don't know anything about the protein, then I would go to detect the peptides first. So I would know that this is a peptide of 10 amino acids, this is 15, this is 20, and then I would do the sequencing on top of that. And after that, I would know the sequence and the amount of that. So I'm showing you those things just for knowledge. There are lots of details I'm not really going through, and structural biology is one of the most complicated domains you would go to. It's really amazing, but it's really difficult. And as I said, I don't see any of you getting to a lab and using mass spectrometry in the first 2 years at least. Now, the third way that we can detect the structure is when we look at the tertiary structure of proteins, looking at how is the final structure, because first, second, and third based on that sequence, and what does it do in terms of, is it a pocket? Is it a receptor? Is it like what exactly the function of that protein? So X-ray crystallography was the first methods being used, and then the nuclear magnetic resonance came after. And both, depending on hitting the protein, if there are some of proteins, they form crystals. So in this condition, we use X-ray crystallography. So you hit that protein and the diffraction of the rays are projected on a film or similar to a slide of a microscope, and then you can reconstruct how that protein looks like based on the projections. So again, this is also very complicated. The nuclear magnetic resonance is similar in terms of the concept, but the difference is that you should have a strong magnetic field around the sample. Now, I'm sure you would say, oh, why he's telling us all these things? And he's saying it's very complicated and I'm not going to use it in my life. This is not entirely true, because you see these 4-5 techniques that we use in a lab to identify a particular protein or a particular peptide of a protein and a sequence of a protein and how the tertiary or quaternary structure of a protein look like. Yes, it helps research to give us a lot of information of what is the function of that protein and how can we modify the function of that protein. But also, the combination of these techniques led to generation of amazing methods to study the human body. So I posted for you here 2 YouTube videos to go and see how the nuclear magnetic resonance has been used. And you know, the MRI is mostly depending on the nuclear magnetic resonance, because when you hit a protein with a radiation and that protein is going to make a diffraction, we are doing this at the level of one protein in the lab. But if you think about it, your body is all of it is proteins, right? And therefore, this is the concept that we're translated into making the CT scans, the magnetic resonance, and the PET scans. So there are 2 videos for you here, and definitely there's a lot of you guys who are aiming to go to the medical doctor degree or to dentist degree, and you will see that small things like this translates into big things like MRI and PET scans and CT scans. Okay, now the last technique I will show you is the cryo-EM. And for those who would really go for research in their life as a career, and they like microscopy and they like studying the structure of proteins, I would really recommend that you go to learning the cryo-EM. It's one of the most powerful techniques and the most needed, I would say. You will be one of the researchers that everybody wants to collaborate with, and it will be very easy for you in the future to get positions. Now, the concept is that you are, again, introducing the sample that you are testing with a pulse of electrons in a vacuum. And then the scattering of those electrons will generate somehow an image. And then there will be a lot of mapping and computation where you get into the level where you approximately get how the protein looks like. So this is, for example, it looks like the pore of the ATP, where in the mitochondria it generates the energy that we need from the mitochondria. And you also could see examples here generating images from bacteria, and there are of course multiple types of the cryo-EM. It's showing you 2 of them. So you can get the details, very, very clean and very detailed details of how the bacteria look like or how a virus look like. So starting from detecting one protein, the techniques are combined together to get you to a level where you can really could see either the whole body, as I told you here in the CT scan, MRI, or PET scan, or you could see pathogens like here. I think it's salmonella. Yes, it's salmonella here. You can see it with 2 different ways, and viruses. So it's very important, those techniques in terms of function. And as I said, go and watch these videos to have more detailed information. Now, when I'm sure at the end of the class, many of you will ask, oh, what am I responsible about these 4 slides? Let this now till the day of the revision. When we get to the revision before the class, I will let you know if these slides will be included in the exam, what exactly will be included in the exam, and what you need to know. So, for those who didn't study molecular biology or genetics, or they didn't go through immune responses, how our antibody react to a pathogen, I have prepared for you this slide. I presented this last year in the pathophysiology course, where it shows you how innate and adaptive immune response work. It shows you how cells present antigens. It gave you an example of hypersensitivity, and it also shows you the function of the complement system, which is really essential for killing pathogens. In other words, every other day we get invaders into our body, and these could be bacteria, this could be viruses, this could be like material that we engulfed, like polysaccharides that came from plants that are toxic. So the defense mechanisms in our body has to process those antigens and present them to other cells in the immune response. So these are the antigen-presenting cells. So they would take those antigens, process it into smaller molecules. So you can see this small dot here is presented on the surface of that cell. And then it will go educate other cell types in the immune cells, in the immune system, in order to activate all these types of cells, either to kill that pathogen or to generate an immune response for long term for that pathogen. So on this side here, you can see that this antigen presenting cell, after association with the T helper cells, it will aid in the education of cytotoxic T cells. And the cytotoxic T cells, now they are educated based on the contact with the antigen presentations, that they can go back all the way to this invader and recognize it based on the surface and therefore kill it. The other route would be probably those antigen presenting cells will educate the B cells, and the B cells are the cells that generate antibodies. So therefore, each time you have an immune response, most probably all these happen together coordinated, where immediate function needs to eliminate that invader if the cytotoxic T cells are able to do that. If not, well, not if not, most probably it's not going to be the case. Then the other arm, it would be just to generate activated B cells, and the activated B cells will generate lots of antibodies. And instead of that cell go directly to need to sequester that invader, it simply sends the weapons, which are the antibodies, and the antibodies are able to sequester that invader. Now, the process is around 7 to 10 days. That's why when you get a viral infection, you would need 7 to 10 days to recover, because the process of taking that virus, processing it down, and choosing a particular part of that virus that is really specific to that virus, but not any other viruses, by the antigen presenting cell, and then dividing of that cell and going to educate the cytotoxic T cells and going to educate the naive B cells, then that takes around 7 to 10 days. And within the 7 to 10 days, you get to the limit where you have antibodies against that. And that's why if this invader comes back in the next few weeks, the antibodies directly react to this one, so you don't need to do all this process. And this is the part of the memory. If the invader came after 2 years, then the memory cells will be activated right away and generate the antibody. And I'm sure you know that you get a lot of vaccines, and that's how the vaccines work. You would take a part of a virus, present it to inject it in the body in a situation that it will not kill you, but your body will make an immune response to that small piece of the virus. And then it will go to these two routes. After 10 days to, I would say 10 days maximum, you will get antibodies, and the antibodies will be generated against that small piece of the virus. You will have a B cell that becomes memory and one B cell that generate these antibodies. And most of the time, one shot is enough. 2 shots is better because then you can reactivate these memory cells. That's why when you are young and you get these shots, they give you a shot, maybe 3 months, and after that, probably 6 months, they reactivate those to generate more of those antibodies. And that's why you get a more robust immune response in case you have a viral infection. So the bottom line, so this is all pathophysiology, it's not related to the course, but I really wanted to stop a minute here to give you a good background about that. Now, what is important for us is nature is showing us how to generate antibodies against pathogens. So we took advantage of this process and we adapted it in the labs so that now, if let's say this is a virus that I never studied, like COVID-19, people try to find a vaccine, then they wanted to generate an antibody super specific to COVID-19, but not to any other viruses that are similar in surface for COVID-19, right? So the idea is the most important step is to identify that antigen that is specific only to this invader. So if you have 3 different invaders, 3 different close viruses, you want to have that peptide or sequence of amino acid. It could be 5, 7, 10 amino acid differences where you want to really generate an antibody that detects only this invader, but not the very close others. So in labs, if I'm studying a protein, not really related to viruses, I'm studying any protein, and I want to really check the levels of that protein, I need to generate an antibody that is specific to that protein. So I will need the DNA sequence, the RNA sequence, the amino acid sequence. I would do comparison between many proteins to make sure that this is the best antigen or sequence from that protein that I will use to generate an antibody. And then I will generate the antibody. Okay? So what are the antigens? And how do we identify antigens based on different types of antibodies? So the definition of an antigen is, in principle, is if you are talking about it from an immunological point of view, an antigen is a protein that is not really expressed in your body. Like in this case, I'm giving an example here, the large polysaccharide, which is mostly on the bacteria. These are foreign from your body, okay? So therefore, your immune system recognizes that these antigens do not belong to your body. Now, if you are speaking about antigens as molecular biology tool, every protein is a different antigen. Like the insulin, you can generate antibody against insulin, and a small part of insulin could be an antigen to that antibody. So the antigens came mostly from microbes. It could be non-microbes like pollens, and it could be on your body. So I'll give you the example of the red blood cells later on. Okay, so what is an epitope then? The epitope is a small part, in other words, a small sequence, short sequence of the antigen that interacts with an antibody. So here you have an antigen. It has the three different epitopes. The first epitope here is bound by antibody A. The second epitope is bound by antibody B. And the third epitope is with antibody C. But since epitopes are small, short amino acid peptides, any given antigen may have several epitopes, and you can see that you have three here. And each epitope can be recognized by a different antibody. So it can be recognized this by a different antibody, each one of them. It's a specificity. Now, lipids and nucleic acid can act as antigens only if they are combined with proteins and polysaccharides. Then, so I described for you extensively how the antibodies are generated in the previous couple of slides. Obviously, the antibodies are in our bloodstream. They are serum proteins. So if you take blood from anyone, you can know what are the antibodies in his. So they are soluble proteins. They are floating in the blood. And they are specifically binding to particular proteins. If you look at an antibody sequence structure, it has a part that bind by complements. So it's called FC because of the complement FC fragment. And it has an antigen binding, AB antigen binding part, which here where it binds to antigens. So the components are really complicated, and how they are generated into heavy chain and light chain, I'm not really going to describe that here. This is more like pathophysiology and immunology. But I wanted you to know that antibodies came in different structures. Some of them are single structures like IgG, IgE, and IgD. These are only two antigen binding sites. It could be that IgA could make a combination between two different antibody molecules, and the IgM could be five antibody molecules.
Excuse me, Professor.
Sure.
Will there be diagrams on the final, like for us to have to like answer questions on?
Could be diagrams, but I will specify from which slides.
So do you want us to know the structures for all of these antibodies here? Or you want us to just know what they do?
No, not at all. So here I'm just showing you the antibody binding, the antigen binding fragment, which is here, and the complement binding fragment, which is there. I'm not going to ask you about the structures of the antibodies. But since you're asking, the next slide is going to give you an example of an exam question. So this is how antibodies work. They can either go and neutralize a target. If this is a virus and you generated an antibody against that virus, then the antibodies bind to the surface of that virus and block its function. They could make different components to make agglutination, and that's what happened when you do blood tests. Or if they bind to a particular protein, they make it, if the protein is soluble, they bring it down so it becomes not soluble. So that's how the three major functions of how an antibody works by its own. Or it can be combined with complement system to make the pore, and therefore it can perforate the membrane of the target, like if it's a bacteria. So that's a combination between complement system and antibodies. Okay, so that I want you to know these. One other example is the blood transfusion. So you know that if you will do blood transfusion or you would donate blood, they will directly check your blood type. And the way they check it is 100, like it's really depending on what antibodies the other person will have. So in other words, if you have a blood type O, then you have in your bloodstream antibodies against A and B. And if you have blood type A, then you have antibodies only against B. And if you have a blood type of B, then you have antigens of A. And then if you have AB blood type, then you don't have any antigens or antibodies in your bloodstream. So in other words, when you say I have A, then the only missing part or not known part is B, and therefore your body generates antibody against B. That's why the people who have the AB blood type, they have all the three different antigens on the surface of their blood cells, and therefore there is no antibodies in their bloodstream against either A or B, and therefore any of these three blood types can give or donate blood for AB persons. On the other side, since the blood type O doesn't have the antigen A and B on the surface of the red blood cells, then there are antibodies against A and B in their bloodstream, and you cannot give them blood except from O. Okay, so this is one of the example of the hypersensitivity. You should have covered this, guys, in the pathophysiology. And this slide is the most important slide for the biotechniques we are studying, and it's going to probably be the last one. First, what is the difference between a clonal and a polyclonal and a monoclonal antibody? So when we are doing experiments or we are going to use antibodies in therapies, we have to make sure what is the type of the antibody. Is it a polyclonal antibody as it looks here, or it's a monoclonal antibody like that? So the way we generate monoclonal antibodies is to make sure we are getting these antibodies only from one single clone of B cells. While when we're generating the polyclonal antibodies, we can generate multiple clones of B cells, and therefore the antibodies are not really specific to only one antigen. So as you see in this image here, this is an antigen. It has multiple epitopes, right? You can see multiple epitopes or multiple peptides that can be detected by an antibody. But when I generate a monoclonal antibody, that antibody detects only one specific sequence or epitope on that antigen, which is in this circle here, the yellow small circle there. So this is a monoclonal antibody. So a single clone of B cells. And only one single peptide out of the antigen is detected. While when we are speaking about the polyclonal antibodies, this means I detected more than one epitope on the antigen. I detected the green epitope, the pink epitope, the yellow epitope, and the blue epitope. So therefore, there is more than one B cells that generate those, so more than one clone. So all the four different antibodies that we see here are against the antigen, but each one of them is detecting a different epitope. So this is a polyclonal antibody. And therefore, the specificity of monoclonal antibody is much higher than a polyclonal antibody, because polyclonal antibodies are a mix of multiple antibodies, while monoclonal antibody is a single one molecule of that antibody. Now with this, I'll just list for you a few of the techniques we will describe, and we will see the applications like the Western blotting to detect the quantity and the specific amount of protein in a lysate. If we want to immunoprecipitate a protein and see what are other proteins that bind to it, like what are the binding partners. If I want to see what this protein bind at which DNA, so this is a chromatin immunoprecipitation, where we see DNA and protein interactions. If you want to see this protein visualizing it in the cell, not in lysates of the cell, like directly in the cell or a tissue, This will be immunofluorescence and immunocytochemistry. Or if you want to sort cells, like I showed you in the single cell analysis by flow cytometry, so you add these antibodies to the surface of the cells, and then you can separate cells based on that antibody. And finally, if you have proteins in the blood or in the fluids, and that's used a lot in clinics, it would be ELISA, which is enzyme-linked immunosorbent assay. Then you can detect proteins or peptides or hormones or antibodies in a liquid sample like blood. So I hope this will give you a good introductory background. And then in the next lecture, as I said, there will be at least 10 to 15 minutes of questions for the exams. And then we will dive into these 4 or 5 techniques later on. And with this, I will stop sharing. And I will stop also recording in case somebody has questions and would like to, okay, where is it?